Ji Bismillahirrahmanirrahim. Welcome to all of you in today's lecture. Today I'm going to explain the concept of SAR with drug development. Concept of SAR with drug development. What is SAR? That is called structure, activity, relationship. Structure, activity, relationship. What does it means really? Once we get a lead molecule, how we get lead molecule? What is a lead molecule? Let me give you a glimpse about it. Whenever we have a library of molecule, whether they're synthetic or natural compounds, we screen them, we check their biological activity, any specific activity. Let's say we have 30 around natural compound isolated from different plant species, and we try them against different bacteria, and we find that out of those 30 molecules, 3-4 molecules have shown good antibacterial properties, so we call them lead molecules. Further, what we could do, we could go for fine tuning of the lead molecules. We can do fine tuning of the lead molecules. Those molecules could be converted into proper drugs. How? After having certain minor structure changes. Why we want to change, why we want to produce, why we want to put in certain structural changes? Because we want to increase their antibacterial activity to the maximum limit, to the maximum extent. Because through the initial screening, we came to know that they have shown, let's say, 40 or 60-65% antibacterial activity. So we find them as lead molecule. We want to play around with its structure, so we can increase their activity. So that will be called as structure activity relationship. We need to change, we need to produce certain changes in its structure so that it become a proper drug. How you could do that? Actually, one could go for minor structural changes in lead molecule. Minor structural changes, okay? What there, I will come to it a bit later. We could go for to change the nature and degree of substituents. We can change the nature of the substituent. Those substituents which are already present in the lead molecules, we can change their nature. Okay, we can make them polar, we can increase their polarity, we can decrease their polarity. Similarly, we can change the degree of substituents. If the lead molecule carries one substituent of halogen atom, we can put in 2 or 3 halogens. Okay, if it carries one hydroxyl group, we can insert number 2 and number 3 hydroxyl groups. Let's see whether increasing the hydroxyl groups, its activity increases or decreases, because the increase in hydroxyl groups would increase their polarity. Okay, they can increase their hydrophilicity. So this is how you can play around. One can play around with the structure of the lead molecule through certain minor changes. What one could do, you can go for the change in stereochemistry. We can play around with the stereochemistry of the lead molecules as well. We can try to generate the stereoisomerism. If the molecule in trans form, we could try to use its to convert into cis form. If the lead molecule is already in cis form, we can go to check the potential efficacy of the trans form. So this is how we can try with the change of the stereochemistry. And so this is how a basics could be done, you know. And what's about the general about the structure activity relationship? What is that? We can introduce ring structures in lead molecule, you know. We can introduce, sometimes we can remove, you know. If the lead molecule already carries any ring structure, 4-member ring, 5-member ring, 6-member ring, we can increase, we can introduce a ring structure, or we could remove the ring structure. Okay. Similarly, we could introduce methyl groups, you know. We can introduce methyl groups in the lead molecules. They could be introduced in two ways. Methyl groups means CH3s in the ring. We could introduce them in the ring of the drug molecule, in the ring of the lead molecule. If a lead molecule carries ring, we could introduce methyl groups into its ring structure, okay? Or we could introduce methyl groups into the main chain. We can introduce methyl groups into the chain as well. Two options, either to introduce those into the ring structure, or we could introduce methyl groups into the main chains. Introduction of methyl groups means we increase lipophilicity. We increase the lipophilicity of the lead molecules. Once we want to increase the lipophilicity of the lead molecules, we could do that by introducing methyl groups either into the ring or into the chain, alkyl chains, you know. Increase of lipophilicity means we are increasing its activity, okay? So it will become more active, it will become more effective, it will show its efficacy. We are going to increase the efficacy of the lead molecule by introducing alkyl substituents, you know. Similarly, we could introduce, or before that, we could introduce, I would like to write it in increase, or sometimes we could decrease, you know. Size of the ring. If a lead molecule carries a ring structure, let's say you have a 6-member ring in the drug molecule. It could be any structure, you know. A number of drugs are available with different structure. I'm just trying to convey you are just a basics about increase or decrease of the ring structure. Let's say this ring, it's a 6-membered ring. One could increase its size, you know. We can made it as a 7-membered ring, you know. The 6-membered ring has been converted into 7-membered ring. Then this new molecule could have a better property, better targetability compared to the previous main lead molecule, okay? Similarly, if the drug carries 6-membered ring, we can increase to 7-membered ring, or one can decrease or make it a new molecule by decreasing its carbons in the ring. Let's say we are using here a ring with the 5 carbons. So increase or decrease in the size of the ring is also an option to play around with the chemistry, with the structure of the lead molecules in order to increase, in order to enhance their biopotential, you know. Similarly, introduction of acidic groups or introduction of basic groups, that could also be done in order to increase the polarity of the molecules, in order to increase the hydrophilicity of the molecules, you know. If we want to increase the hydrophilicity of the lead molecules, we can go to insert acidic groups, or we could put in basic groups. These groups are also be used to, if we want to generate hydrogen bonding in the lead molecule. If we want to introduce a group which has the ability to develop hydrogen bonding with the target molecules, so what one could do, we need to have some groups in the lead molecule in the drug which can have the ability to develop hydrogen bonding, you know. So these molecules have the ability to develop hydrogen bonding when they come when they come in interaction with the targets, mean they come closer to biological molecule, they come closer to some activity cavities of the enzymes. So they will have to develop their binding with the target. That could be through electrostatic interaction, that could be through covalent bonding, that could be through hydrogen bondings. Okay, so if a drug is going to attack its target through hydrogen bonding or electrostatic interactions, means we need to have some acidic or basic components introduced in that lead molecules. So this is how we could play around with the chemistry of the lead molecules by introducing acidic and basic groups. Similarly, we could introduce halogens. We could introduce halogens into the lead molecules, you know. They're also basically done for the development of hydrogen bondings. Okay, such groups are introduced either into the ring or into the side chains as per the requirement, as per the structure of the target molecules. Okay, so these are the few of the areas which you need to know, that how a lead molecule could be converted into a drug molecule, you know, by doing minor structural modifications. We can convert a lead molecule into a drug, into a pharmaceutical candidate, into a pharmaceutical product which has the best possible biological activity, whatever we are looking for. Let's say we are trying to develop an antibacterial agent, or we are trying to develop an antiviral agent, okay, or we are going to develop an anticoagulant agent. So depending upon whatever is our requirement, we can play around with this structure, with this chemistry. So let me explain all these facts with one very relevant example, you know, of the lead molecule. So I can show you how the lead molecules could simply be used for such, chemical transformations for the preparation of. Let's say I'm going to draw a lead molecule. I've taken it from literature. So many lead molecules are known around, so this is just to recapitulate what I've just explained to you guys. So let's say this is one of the lead molecule which has been obtained from, which has been isolated from plant species, and since let's say this compound has shown promising antibacterial capacity, antibacterial potential. So now we want to check, we want to increase, let's see whether minor change in its structure can increase its activity potential. Let's say one can go to introduce a methyl group over here. Okay. In the next step, what one could do, we could introduce, let's say, this sort of a compound. Okay, it also carries hydroxyl group as well. So we could introduce an halogen atom there. In any other next step, in next transformation as well, we are going to develop another lead molecule where what we are going to do, let's say we replace this hydroxyl group with thionyl, this thionolic group have been introduced over here. So this is how the lead molecule we are going to use it. Okay, or this is an acidic group, this is a basic group. What one could do, let's say we can introduce over here some sort of component. Let's say we have introduced a simple this is a compound, or we can develop another component, we can develop another compound which carry, okay, or I'm returning back to the original ones. Let's say, okay, simply one could go to introduce a ring structure. Okay, and then we can go to check its efficacy of its potential, whether it has been increased or decreased. Next time what one could do, we can try to introduce this ring structure at this side, Again, it's up. We can one time, let's say we can introduce structure on over here. We can, next time we can try the other way. A controlled reaction could give us the insertion of the ring at this carbon. Okay, so there are so many possibilities. What I'm trying to give you a glimpse that a lead molecules, once we receive them from natural resources, or we prepare them in the lab and we see through initial screening that the compound has shown a promising biological activity, then we can play around with its structure. We can introduce the rings I have just shown. We can increase, we can decrease, okay, the size of the ring. We could introduce methyl groups in the chain. We can introduce methyl groups in the rings. We can introduce halogens, Sometimes we can try with chlorides. In halogens are 3-4 different types, so we can try with one. Then in another lead molecule, try with fluoro. We can bring in chloro, we can bring in bromo. Maybe chloro is the lead molecule with chloro that shows best activity compared to the other molecule which has bromo or fluoro components, So this is how, similarly, let's say it's a one acidic group. It has one basic part as well. Let's say maybe introduction of another acidic group could increase its, hydrogen bonding capacity. It will also increase its water solubility. It will also increase its hydrophilicity. Generally, the drugs are supposed to be more lipophilic than the hydrophilic. Okay. However, insertion of especially SO3 groups SD groups, they increase the excretion of drugs from the body. The drug finally has to be excreted from the human body. So in order to increase the excretion of the drug, generally the SO3H groups are introduced to enhance their excretion from the body. So this is how the lead molecules, which are found promising candidate after the initial screenings, they can be moved to R&D section in the pharmaceutics or in the labs in the universities. Then being pharmacist or being student of the pharmaceutical chemistry, having background knowledge, how you can introduce substituents on the rings, how you can introduce substituents on the chain, how you can introduce SD groups, how you can introduce basic groups. That is the main requirement to achieve in order to convert lead molecules into proper drugs. So what is required? Being a student of pharmacy, of pharmaceutical chemistry, you need to learn the basic chemistry, how the alkyl substituents could be introduced at the ring, at the alkyl chain, how rings could be introduced, how size of the ring could be expanded, how the size of the ring could be reduced, how hydroxyl groups could be inserted into the rings, and how the hydroxyl groups could be inserted on the chain. Similarly, how halogens could be introduced. So you need to have basic structure, basic knowledge in the chemistry that is very much required for the conversion of lead molecules into the drugs. So this is what I'm trying to give you an intro, a glimpse about the SAR and drug development, structure activity relationship. This is what I'm trying to convey. This is what I'm trying to convey to you today, that the lead molecules could be converted into drugs with minor to major changes in its structure. Introduction of rings, removal of rings, introduction of hydroxyls, removal of hydroxyls. Once, if you are intended to increase its lipophilicity, or you want to increase its hydrophilicity, so you can go the way you want by using your knowledge in basic knowledge in chemistry. So you need to keep an eye about the basic conversions in organic chemistry. That is very much applicable for the synthesis of drugs from the lead molecules. I hope you got the message. Thank you very much. I will give 1 to 2 more lectures on SAR, structure activity relationship. I will bring in 1 or 2 more relevant examples. Let me give you one more, relevant examples of the drugs, how they have been developed from the lead molecules. I will bring those today. I'm just trying to give you a glimpse about playing around with the structures of the lead molecules. If you can remember, it's a salicylic acid. It has been converted into acetyl salicylic acid. Salicylic acid, a minor changes acetyl salicylic acid, and it has become aspirin. A very much frequently used medicines, aspirin. What has been done? A simple change in its structure has given you an active compound, active pharmaceutical products. So this is what I'm trying to convey. In next lecture, 1 or 2 lecture on SAR, I will bring in those drugs which has been developed from lead molecules, what sort of basic transformation has been done, what sort of basic substituents has been changed, what was the starting lead molecule, and what sort of the structure modifications was done, stereochemistry was changed, hydrophilicity was increased, hydro lipophilicity was increased, how it has been done, and what are the final drugs. I will bring one to two those examples to explain and to polish your concept about the SAR structure activity relationship. So thank you very much for your attention, please. Assalamu alaikum.